Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Rudi Appels. He's an honorary professor at the University of Melbourne as a research fellow at AgriBio that comes out of Trobe University. He focuses on the wheat genome. I guess the wheat genome is vastly more complicated and it has a lot more base pairs than the human genome, which is kind of strange. But we'll get into that. And Rudi, welcome. How you doing?
Yeah, I'm very well, thank you. And as I said, it's a pleasure to be able to talk about some of my work with you. Yeah.
Well, wheat, I guess, is is very important because so many. It's harvested, all over the world. I guess especially in the United States. So it's very popular. But I didn't realize this genome is so complicated. So how did you first get interested in wheat and its genome? And we'll go into that.
Yeah, the wheat genome. There's most of us have got pairs of chromosomes, and that's called one. Like we've got the pairs of chromosomes in our human cells, and that's sort of a genome's worth. The wheat genome has 3 sets of genomes, so that makes it really complicated, and we'll get into that a bit later. So we have the A, B, and D genomes. So I got into wheat long time ago now. I'm a biochemist and chemist by training, and my natural inclination is to visualize molecules and what they do. And how I got into wheat initially, it's a bit tricky going back 50 years to try and see what I did well and what I thoroughly messed up. But I think the sort of key part for me was when Jim Peacock in CSIRO Canberra gave me the opportunity to work on wheat. I was a research scientist there in 1977, and it was a real this is where I want to be type of period for me in my research. Although this is way before the technical and computer capacity we have today, I still managed to isolate classes of DNA sequences that were able to specifically track Y chromosome segment into wheat. And this was like something that really turned me on. It was like cloning way before the days of cloning.
Well, so, what does that mean? You were able to track Y sequences in wheat? Does that mean that rye was a progenitor of wheat and you could see the breeding, or what do you
No, this is really interesting, Richard. In the 1930s, people were doing crossing between rye and wheat, which was really hard to do. Doesn't happen all the time, but they pushed it and they made a new crop called triticale, which was really interesting, and 50 years ago was being pushed as the new cereal for consumption in biscuits, particularly in Canada. So triticale had the rye chromosomes plus the wheat chromosomes, so it was a real thing. And so when you then back cross wheat with triticale, you get bits and pieces of rye, and it's basically like cloning. But you're cloning big segments. Yeah, so that's, and it just fired me up to be able to look at something specific, like a rye segment in this case.
Well, what would be the benefit of crossing wheat and rye? What properties were people trying to breed into it?
Yeah, so yield, large grain. The triticale has a very large grain, new disease resistance, and there was some sort of, it just tastes different as well. So it's never really taken off as a new cereal, but it was it came pretty close. It just I'm not exactly sure why it never really got people's imagination, but there were sort of the big advantages were for the growers that they had improved more stable yield, which probably may have been where that fell down a bit later, and then also the taste, which was a very nutty sort of taste, which was pretty good in the sort of biscuit type material that the Canadians were producing. Yeah, so it's the two aspects were of interest. And I think with wheat, Richard, it's sort of really interesting how it's such an ancient crop, but it's been used for crossing everything to it. And I guess rye was one of them. And then you have all these other wild relatives of wheat that get crossed into it for different traits. It's usually disease resistance.
Okay. Well, the wheat that's grown nowadays, is it very self-similar, or only, is only one variety used, or, to make it more robust against disease? If you look at a given field, are there many different types of wheat, or is there too much of a trade-off with having different types in the field?
Yeah, so if you look at one field, it's pretty much a uniform crop. Like it's what they call a monoculture. But worldwide, there's a really widespread varieties of wheat, and in different continents, they have different sort of what we call alien chromosome segments. They're the bits of crosses that were done by breeding to introduce resistance. And so that's given a lot of variation in the wheat varieties, which are then grown, and they adapt to different environments. Even in the North American continent, you have the wheat grows in the really cold climates of Saskatchewan, but it also does well in the Midwest. So, it really has adapted. And that means you've needed to have different genetic material inside the wheat.
So the wheat genome, how many base pairs does it have? I've heard it's just like a huge genome. And does it have chromosome pairs, or it has like quadruplets? What's unique about it?
Yeah, so it's got 16... We estimate now with the sequence, it's got 16 gigabases, so that's 16,000 million bases. The human genome has got about 5 gigabases, so it's complex. And the bread wheat, as we normally have it, has got this what I mentioned briefly before, the A, B, and D genomes. So these are the sort of reflections of the progenitors, the way the wheat evolved from diploid, like when there were just 7 pairs of chromosomes, and then one set of 7 pairs of chromosome combined with another another wheat line way back, and it formed like 2 sets of 7 pairs, and then finally about 10,000 years ago, just in the Caspian Sea area in Iran. There was a 3rd set of 7 chromosomes were added. So all up, there are 21 pairs of chromosomes, but they're sort of in these sets of 3, the A, B, and D genome. And so in the 10,000 years since some farmer would have been looking through his fields and he found this really interesting large grain in his field of normal sort of what we call pasture wheats, the A, B genome wheats, and this was then the first A, B, B, D, D, the 3 sets of chromosomes, and then somebody must have been really observant 10,000 years ago and kept the grain, nursed it along, and finally it became the bread wheat, which is really, it's fantastic because with the 3 sets of pairs, 7 chromosomes, so a total of 21 chromosomes, there's just a huge flexibility. It's just an incredible crop. So you can, it's just very, very dynamic at the genetic level.
Can you tell how many rounds of breeding have occurred by looking at ancient wheat to wheat nowadays, and maybe even the last 100 years? Has there been a lot of changes and a lot of selective breeding, or has it been relatively few? If you look, does anyone look at the history and track it?
Oh, yeah, very much. So this is traditionally, there's been a good source of argument to say, well, what was the exact history of wheat? No, that's a very big area. But in the last 100 years, for example, just to answer your question, there's been this really big burst. I mentioned in the 1930s, there was this effort to cross rye with wheat. So similarly, all these other grasses which normally don't cross to wheat were forced in natural crosses, but they still had to be nursed in the lab, what we call wide crosses. And this way, alien bits of chromosomes have been introduced into wheat. So if you now look at what we have in our hands. There's this incredible history of what we call alien chromosome integration. And as I said, it's mainly for introducing new disease resistance, because that's a really never-ending battle to grow wheat in such a hostile environment as the world is with disease. And yeah, it's that you can see that history now in the genome. It's an absolutely amazing story that what we've as humans have developed in the wheat. Yeah.
What does your work on wheat look like today? What are you trying to accomplish or understand?
Yes. Okay. So this is a big question, and I think there are a couple of things that are really are standouts. I guess if you look back at what the DNA success story has been, is that like specific genes have been obtained, and we can see them, select them, and have a diagnostic for them so that we can then do the breeding more efficiently. And that's now being pretty much adopted. And I've got some examples later if you want. And I think so you're really asking me for what is the vision, I guess.
Even if it's multiple elements. I mean, start with one and let me know. Is there, again, just just break it down into pieces. It's okay.
Yeah. Okay. So with so on the one hand, the wheat genome, if you like, it's so complex. It's a life in its own, and working out how the gene networks are expressed and cooperate with each other and are modified by the environment are really important. There's a lot of very specific. So that's the big picture, right? The general scope of how genes work and produce a good wheat crop in a particularly hostile environment. The more specific thing is for areas like the people react badly to some of the wheat proteins. That's what people call it, gluten insensitivity, and that's an area I think which is more specific, where I think we can, now that we know all the genes involved in the flour proteins complement, that we can actually start to really work with people who have this and look at what some of the issues are. In one example, again, just as I like the specific things, there's a group of chemicals, the herbicides, which you can have resistance for. These are called imidazolinone. And I think one of my things that I did, which I'm particularly proud of, was that we discovered, we characterized a new mutation, and it was a new gene for this imidazolinone resistance, which has been adopted. So I think there are specific examples where being able to identify a mutation and then track it in a breeding program to then add to the valuable part of what is in agriculture. And then the other side is the human health.
So when you say wheat is dynamic, does it tend to change on its own? Does it undergo like epigenetic change or even genetic change on its own? Does it breed on its own, or is the way it's planted it just will grow and go through part of the life cycle, but not go through a stage where it crossbreeds? Yeah.
So Richard, wheat wouldn't exist without human beings, and probably human beings wouldn't exist without wheat. So you can't think of wheat doing something on its own. It's all driven by humans. There's a real sort of intimate relationship there that you can't separate. And so what drives the change in the wheat genome is when we as humans take it to environments which it's not normally suited to, and there may be some dry conditions or really cold conditions like in the northern hemisphere. So those, when wheat is what we call selected to grow there by humans, it then is coincident with the genome change that's occurred. So there's basically a lot of variation sort of sitting there, and the humans come along and pick the winners, and they may have a particular rearrangement of the DNA, or there may be a deletion which was just sort of hovering in the background. But in this particular instance of this environment, it then gets selected and it becomes the prominent feature of the wheat genome. So yeah, it's really a situation where the human selection is the real driver for selecting variation, which is sort of sitting in the background, I guess you could say, and it's sort of there, but then we select it to make it prominent.
Well, why would different types of wheat do better in different areas? Is it just, does climate have a huge effect on the different types of wheat that are planted? And if so, what are the effects?
Yeah, they're huge. If you think of the frost tolerance that's required when you grow wheat in you plant it before the snow comes, and then it sits there during winter, and then in the following spring it pops up and gives you a good crop. That's an entirely different scenario, if you like. It needs the plant to be frost tolerant. It needs persistence through the winter while it's sort of, it has to basically persist until the next spring. Compared to in Australia, you plant your crop and it sort of coming up around this time of the year, it has strong sunlight, hopefully some rain. We don't get too much rain. And then come October, it's then harvested. So it's a completely different, in Australia, the plant is, it's planted, and with the first rains that come for the season, it goes for it. It grows like mad, as opposed to in the snowbound countries, it's planted, it sort of starts to germinate and then sits there while the snow covers it, and it hibernates, and then next spring. So there's this really completely different requirements for the plant to survive in those conditions.
What are some of the surprises that you see in the wheat genome? Can you tell what can be exploited from here? What can be modified from here to get certain effects? Like what are wheat's biggest problems right now? Are there diseases that are targeting it more than others? What are some of the issues surrounding wheat right now?
Yes, I think disease is a never-ending problem. I think there's no question about it. It's the big issue. So that's a huge issue. It'll never change because the pathogens, the fungi, the root and nematodes, you name it, they're sitting there wanting a free feed on the wheat plant. And there's always the challenge of getting genetic resistance, because the genetic resistance is cheaper to implement than a chemical version, and more environmentally friendly as well. So that's clearly a big one. I think the emerging issue that I think people are really talking about a lot are this environmental problem, the global warming. It's got everyone's attention, and I think there are some varieties. It's sort of a bit hard to explain this, but because we don't understand it. But there are some varieties that seem to be able to kind of do what winter wheats do and hibernate. But so in Australia, there are some varieties that can sort of, when the rain is not coming, they're much better at sort of sitting there in a mini sort of hibernation until the rains come, and then they go for it. There's that sort of what we call plasticity in that growth habit. That is something which would be really cool to capture if we could understand the signals that turn off, if we could say turn off the crop properly so it doesn't waste resources when there's no rain in our case. And then when the rains come, it switches back on and it goes belts out a crop. So I think that sort of what does this really mean is that getting a wheat crop that's engineered or selected to be more in tune with environmental than the continually changing environmental conditions that we grow it in. And I think that's it's a lovely, for me anyway, it's a beautifully complex issue with the whole plant genome expression being involved, and there's a lot of issues. So that's sort of superimposed on the need to be able to grow the crop in a very hostile environment when it comes to disease. And So those are two layers, and then the other layer is our human consumption requirement, which is in terms of the gluten sensitivity. And so so those are the three broad areas, if you like.
So I don't know, is there anything in the genome that you see of wheat that, again, is just very strange or unusual or unique perhaps only to wheat that holds, I don't know, some mystery to it that, really needs a lot of study to understand?
Life is pretty uniform in terms of the way it works. I think we see a lot of things in the human genome, the very sophisticated work that's done in the human area, which applies to wheat, and it's really just to capture that knowledge and see how this works in wheat. I think there are some very specific things, but they're not unique to wheat. I think there's the cereals, there's barley, maize, and a lot of those. There's a lot of common ground there. I think the thing about wheat is it's what we call polyploid nature. There are three sets of genes for everything, three sets of pairs. It's a hexaploid, which really introduces a whole new ballpark of rules of the way genes interact, which probably is not possible to look at in other genomes, and so I think in other cereals. So I think that's probably the really unique part, which gives us a complexity which we really, I guess, keeps a lot of us wheat people happy. And it's that flexibility of that DNA space, which is the thing that is unique, I think. I've said this a few times now, but the amount of variation that's been selected by people, by humans over the last millennia, it's really it's amazing, and it's accelerated in the last 100 years when we've had that very intensive crossing with the wild relatives.
Very good. So what do you think that is going to be a breakthrough in understanding of wheat over the next year, or is this really going to be a slow process and it's going to be years before there's any major changes in our understanding of it?
Yeah, I think, Richard, in my mind, I'm obviously biased, but I think the really big breakthrough was really, in a way, was characterized by a recent publication in Nature, I think it was, that where it's become quite evident there is a suppressor. A lot of things are suppressed in wheat, and like in humans, a lot of nature is spent stopping things from happening. Like if you don't stop cells from dividing and they'll run free, in humans you have a cancer. So it's that sort of ability to suppress things which is very strong in nature generally to produce an organism. And the recent example that was done by, like as always, a big collaboration from people across the world, was to demonstrate that this one gene on the D genome, so it's one of the genomes, had a gene that actually suppressed resistance genes on the A and B genomes for stem rust. Okay, so this is really, so the ability to resist a whole range of fungi is there, but it's being suppressed for whatever reason. And so when you actually then mutate that gene that's causing the suppression, you get a whole new range of disease resistance. And so that's what, and then there's been that really interesting development where it's part of the interaction, the three-dimensional interaction inside the nucleus of that D genome with the A and B genome. So, or the A genome actually in this case. So you had it's that sort of complex. You have to sort of try and for me it's like a universe out there. You've got a lot of genes that are there, and they're the equivalent of stars for me, and they're all interacting, and you can see galaxies. Well, you have inside the nucleus, you have in a way a congregation of the genes, the stars, into a bigger superstructure I think of as a galaxy. It's just a personal view where they're interfacing each other. So it's a three-dimensional interaction which we can now measure using the technologies that have been developed in the human area. Yeah, so I think just to summarize what I'm trying to say, not explaining it all that well, but the new sort of area that's really interesting is that sort of shaping of the three-dimensional structure of the nucleus that then controls the expression. And this example of the suppressor of the stem rust resistance is one good example which involved this sort of interaction.
I mean, just give one real specific example. How would the 3D structure of the genome affect its expression? I've learned about epigenetics. A part may be methylated and then therefore inaccessible, or this is in people. I don't know if this goes for wheat, but how does structure govern the ability to express parts of the genome?
Okay, so that's, I think, part of that, if you like, that three-dimensional thing is that it affects the way DNA is then modified. There's, I really don't think that we really understand that properly, and I think that's the new area. But the epigenetic changes are basically when you chemically modify, you methylate inside the cell, you get methylation of the DNA, and that can then switch off genes, or when you demethylate, it can switch them on. And so that's if a gene is located in an area where there's a lot of methylation, so it folds into what used to be called heterochromatin. If it folds into that, then the genes in that section will get more methylated, epigenetic type modification as you want to look at it, and that then controls the expression of those genes. It's how a three-dimensional structure can fold a set of genes that are normally active into an area of the nucleus that's where there's more methylation going on, and that they then get switched off. So that's the sort of interesting modification, if you like, of the gene activity at that sort of the level that you're asking about, is the epigenetic level.
Okay.
Yeah.
Well, very good, Rudi. What's the best way to make this accessible to people that are listening, so they can get a grasp on the nuances of wheat and why it's so special, why it's so amazing? What do you recommend they go for a primer on what we've been talking about?
In terms of reading about it, is that what you're asking?
Yeah, right. What's the next step for a listener that wants to know more, about wheat, the history of it, its genome, the challenges it faces, etc.? Where can they go for an overall view of things for a start?
Yeah, I think you gave me quite a good primer. It was Lucian did. I thought that was really good. It was I'm not exactly sure where this came from. So sorry, I haven't got that at my fingertips. But
it's okay.
It was
we'll edit that out. No problem.
Yeah. So it was really it was just a discussion that was. It's only a one-pager, right? It's sort of like and it was a discussion on wheat genome that was released in August 2018. So there are I've been noticing there are some really good primers, as you call it, which are being put out, which can give people a feel for, I guess, the excitement for what's going on in wheat. And I think the sort of thing we take for granted when we go for a holiday is we get out a map, we use Google Map, and we drive to a destination and we know where we are. Well, it's that exact same analogy for the wheat genome. The Google Map of the wheat genome is what we now have, and we know where we're going. It sort of lets us map things where any one gene is located in a particular stage of development of the wheat, and it gives us the chance to then start to grow wheat in environments that are really always we're battling to get enough.
So what's the best way for people to find out more about your work and your specialization? How can they follow up?
Yeah, I think when I Googled myself, and I think that comes up with a fairly comprehensive of who I am with publications. I think probably the simplest way is to look for anything with the International Wheat Genome Sequencing Consortium, that reference to it. is probably the best way, because the IWGSC is very active, and it still has a really terrific director, Kelly Eversole, driving that. And there really are a lot of communications there, and sort of now. So that's really the answer to your question. I think the IWGSC has a lot of communication material, and if you Google the IWGSC communication, I think that would be probably the best source.
Very good. Well, Rudi, thank you for coming on the podcast, and I appreciate it.
Yeah, good. Okay then, great.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.